Hello, welcome to our 2021 Lab Root series sponsored by Thermo Fisher Scientific. My name is Leah Lavery, a marketing manager for Life Science Electron Microscopy and your moderator today. This is the second webinar in our series, and we welcome back any returning attendees and a big hello to any new attendees wherever you are in the world. Many of you know Thermo Fisher from other life science areas, but I want to introduce you to the Life Science Electron Microscopy division. Our Materials and Structural Analysis division provides complete workflows from cryo-electron microscopy, structural determination of macromolecular complexes in native states, to 3D reconstruction of tissues and cells. Last year, we broke the atomic resolution cryo-EM barrier with the introduction of the Selectris imaging filter with 300 kV cryo-EM. Today, we will be discussing what's possible with the energy filtering for 300 kV and also 200 kV cryo-EM and several examples. We will highlight the discoveries of what's now possible to look at underlying protein function and cellular processes with the Selectris imaging filter. Today's presentation is titled Cryo-Electron Microscopy, Membrane Proteins Down to Atoms. It will be given by my colleagues Marc Storms and Abhay Kotecha, and our special guest speaker, Professor Stephen Brohawn from UC Berkeley. After today's presentation, there will be a question and answer session. Please submit your question at any time through the Q&A window. Let me now introduce my colleague, Marc Storms, Product Marketing Manager, to start us off.
So welcome everybody to this Thermo Fisher UC Berkeley webinar. Thank you once more, Leah, for introducing us. I'm very excited today to show you again some new data and new developments actually that we have been working on the last couple of months or years actually. And while showing this video of the cryo-C4, you can very nicely see the interior of the system and what is really inside the cryos, and also the components that we talk about today, and I will shortly recap, are all inside this box, as you know. So for those who have been listening before to this webinar cycle, we have been discussing a lot of new innovations recently, actually the last couple of years already. First of all, as a short recap, of course, we have the cryo-C4. You already saw the animation on the previous slide, and of course, we also have the glaciers. I think both tools are famous for their significantly reduced height, which makes it relatively easy to install the systems in a standard lab room without too significant room renovation efforts, basically. With respect to both platforms, we've also implemented significant automation, not only automation in order to get to the right optical status before you start doing an experiment, the so-called APM or Advanced Performance Monitoring, but also automation in the application software, for example, EPU, which makes it easier to facilitate, for example, the screening of grids. And last but not least, of course, we have been pushing the productivity by implementing, at least with respect to the platforms, aberration-free image shifting and fringe-free imaging, as you all know. The second actually branch to the tree, if you like, was introduced last year, the Selectris, which actually is our newly designed and developed energy filter. It's very famous for its stability. I will spend on the next slide a little bit more time on this. It's also actually very easy to use in terms of setting up the experiments. And in case actually there would be some tuning needed, because of course there is always actually some alignment needed, and this is actually going to happen very easily and then very easily, because it has been implemented, this process, into our application software, EPU and tomography. But I will get back to that on the next slide with a little bit more details, especially when we talk about the stability. And then the third component, which is of course also an integral part of the Selectris, but it can also be used, of course, standalone, is the Falcon 4 camera. We have significantly boosted the speed of the Falcon 4 camera, but in addition to that, we have also pushed the DQE, the sensitivity, across the entire full frequency range for those doses that are typically used for cryotomography and single particle analysis. But last but not least, we have also implemented this nice feature called electron event representation, which actually is a clever way to do more efficient lossless data compression, while still benefiting from the full temporal and spatial resolutions. So we can also actually enable, because of this sub-pixel accuracy capability, the super resolution capabilities of the camera. And now I'm pleased actually to announce the fourth addition to this combination. That's why we talk about the power of 4 actually in this presentation. And that's our ColdFAC, which we call ECFAC. The E stands for energy spread, because we've optimized the energy spread performance of the ColdFAC. And as I will show you also in the next couple of slides, this ColdFAC enables to get higher contrast, push resolutions, especially beyond the 2 angstrom level. And it also gives us the possibility, and gives you the possibility, to actually achieve resolutions in a shorter time frame. But first, a little bit, again, some extra information with respect to the selectors. I already mentioned that this is actually a very stable energy filter in terms of the zero loss position and behavior over a longer period of time, which of course is important if you want to do long enduring experiments. And in these 2 graphs, you show actually the actual measured performance of the selector stability on the cryos and on the glaciers. And you see that the behavior, the measured behavior of the zero loss position is very well within our specification of plus or minus 1.5 electron volts if you measure over a period of 24 hours. In fact, it's more close to plus or minus 1 EV in reality. So it's extremely stable, and the small periodicities that you see here as a small dip actually in this measurement are related to the autofilling of the liquid nitrogen. As I mentioned, it's actually a very stable filter. So in case you have to do some frequent centering and tuning, it happens invisible, because it's been fully integrated into our application software, and the fully automated filter alignments are also done relatively fast. Within less than 5 minutes, you've really done it. The most actually practical value, again, is the fact that we have this very stable energy slit, and that basically is a key enabler for very exciting scientific results that will be discussed by Stephen and Abhay later in this presentation. And that brings me to the ColdFAC. As I mentioned before, our new ColdFAC is optimized for low energy spread. That's what the E stands for, the ECFAC, or energy spread optimized ColdFAC. And as you can see in this graph, where we have been comparing the ColdFAC behavior versus the classical XFAC behavior, then you can really see that the energy spread has significantly been improved. Our official specification here is less than 0.3 electron volts compared to the XFAC, the high brightness XFAC, which was actually the standard FAC on our cryos, and glaciers that has an energy spread of 0.7 eV. And the advantage, obviously, of having this better energy spread behavior is what you can see back if you look to the contrast transfer function, and you see that especially at the high spatial frequency range, there is a much stronger contrast signal when you talk about actually the lower energy spread behavior. And that, of course, translates back into higher resolution capabilities, and Abhay and Stephen will actually talk about that in this their presentation parts. And also, because of the better contrast behavior, you can reach actually higher resolutions in a shorter possible time. With respect to the other specifications, like the brightness, they are similar to what the XFAC specifications are. So we have a very high brightness actually, but simultaneously we have a very low energy spread performance. Spot drift also same specification as for the XFAC in this case, and the irregular flashings, or the regular flashing actually intervals that you have to do every once in a while, is also something that doesn't disturb your experimental performance, because it's fully automated and embedding into our EPU and tomography software. So you will not be actually bothered by any of these interruptions with respect to your experiments. And last but not least, our ColdFAC actually has a 5-year standard warranty to actually show the confidence that we have in the product. And with that introduction on what we call the power of 4, I would really like to show you and introduce you to Stephen Brohawn, the first speaker, and then Abhay will follow up, who will talk about what you can really do with the combination of these powerful instruments. A short introduction, Stephen actually is Assistant Professor of Neurobiology at the University of California in Berkeley at the Department of Molecular and Cell Biology and the Helen Wills Neuroscience Institute. Their institute is basically focusing, and also his work is on understanding the basis of sensory transduction and electrical signaling of the nervous system. And to this end, actually, his lab is using cryo-electron microscopy as a major tool to investigate the structure-function relationship of membrane proteins and also ion channels. Important to mention is that Dr. Brohawn is a New York Stem Cell Foundation, Robertson Neuroscience Investigator, and his work has been recognized by at many occasions already, for example, with the Sloan Research Fellowship, McKnight Neuroscience Scholar Award, and the Klingenstein-Simons Research Fellowship. So we were very happy to have Stephen here today in our webinar series. So I give the floor to you, Stephen.
Okay, thank you very much, Leah and Mark, for the introduction and the invitation to be here today. I'm excited to tell you about two short stories from my lab about improvements in resolution of the structures of small membrane proteins using the instrumentation that we're discussing here today. And the best of these reconstructions is shown in the background of this slide here at 2.1 angstrom resolution. So the first of these stories concerns a protein called ORF3A. This is a putative viral ion channel encoded by the SARS-CoV-2 genome. We became interested in this protein at the beginning of the pandemic last year as a potential alternative target for vaccine or therapeutic development based on work that had been done in the related SARS-CoV-1. And we're also interested in this protein because it appeared to represent a novel membrane protein ion channel fold. And so this is a very small protein at 31 kilodaltons as a monomer. And so was a challenging target for cryo-EM, but a fantastic postdoc in my lab, David Kern, was able to, in just a few short months, develop an expression purification system, reconstitute dimeric 3A into lipid nanodiscs, and on instrumentation here at UC Berkeley, collect data and determine the structure of this protein to 2.9 angstrom resolution. And the local resolution of the final map is shown on the right of the slide here. So I'd just like to acknowledge Jonathan Remus and Dan Tasso at the UC Berkeley Cal Cryo facility for coming in at a tough time in the pandemic to collect these data. And Chris Hull, a graduate student in my lab who worked with David to process the data and generate this reconstruction. And so we were able to learn quite a bit from this really rather high resolution map for such a small membrane protein, and posted a preprint in June of last year describing the results. But we were even more excited to have the opportunity to collaborate with Abhay Kotecha at Thermo Fisher to see if we could use this new microscope hardware to push the resolution of this reconstruction even further. And so we took the grids out of the Arctica that we collected the first data set on. Dave crossed his fingers and shipped them to the Netherlands, where Abhay was able to collect a data set for us, and that's shown here. And indeed, from these data, we were, in fact, able to improve the resolution of this reconstruction substantially to 2.1 angstrom resolution. And so this was using the Titan Krios with cold FEG and Selectors X energy filter and the Falcon 4 camera. So you can see immediately, I think, the improvement in map quality from the image shown on this slide, and I'll just show the two maps next to one another so that the comparison is more clear. It's really substantially more detail in this higher resolution map on the right. I put this table here just to emphasize that these data were collected similarly. In fact, it's a smaller data set in the higher resolution that led to the higher resolution reconstruction, about half as many final particles. And I also wanted to point out that all of these data are publicly available. The raw movies, the maps, and the models are all deposited. And we were excited to see both the EMDB and the COOT software package use these data in their recent modeling challenges. So we were able to do 4 additional things in this higher resolution map compared to our first reconstruction. First, we were able to model a portion of the membrane scaffold protein, this MSP1E3D1 protein in this kind of tan color on the map on the left here. Second, we were able to correctly model a large number of side chain rotameric positions that were ambiguous in the 2.9 angstrom map, but clearly defined in the higher resolution reconstruction. Third, we were able to model approximately 120 ordered water molecules, which are shown in blue in the map and the model here. And fourth, we were able to model 2 specifically bound lipid molecules inside the 3A central cavity. So we see that 3A adopts a novel fold here with 3 transmembrane helices and a cytoplasmic beta sandwich domain that assemble tightly into a dimer. And as we were thinking about the putative function of this protein as an ion channel, we observe that 3A adopts a structure in which it has a large inner cavity that's polar, that's connected to the cytoplasm and the surrounding membrane through 3 pairs of water and lipid-filled tunnels. And I'll show you those in a little more detail here. So the first set of these tunnels is shown on the left in these slice throughs of the surface of the protein. There's a upper tunnel, pair of upper tunnels, which we speculated were connected to the surrounding lipid bilayer. And indeed, in these tunnels, we don't see evidence of bound water molecules, ordered water molecules. There are a pair of lower tunnels that are, we speculated, were connected to the cytoplasmic solution. And in fact, these tunnels were lined with ordered water molecules, consistent with that idea. And on the other side of the slide here, you can see a third pair of tunnels, these intersubunit tunnels, that had density in the 2.9 angstrom resolution reconstruction that was difficult to model, not really interpretable. But in the higher resolution reconstruction, we can see very clearly that these densities correspond to ordered lipid molecules that are bound inside the channel cavity. And we modeled these as a pair of DOPE lipids with the ethanolamine head group of the lipid pointed inside the channel cavity, making interactions with very conserved residues within 3A. And so this improvement of resolution has helped us think about how this protein might function in the viral life cycle as an ion channel by adopting this fold in which a large polar cavity is connected to the cytoplasmic solution through these lower tunnels in which ions can freely pass, and then potentially traverse to the other side of the membrane through a central pore that's closed in this structure or along partially exposed hydrophilic grooves on the outside of the protein. Okay, and so the second story that I'd like to tell you about is the following. Concerns a ion channel called TAS2. It's a potassium selective channel in eukaryotes that's pH regulated. And it's one of the conserved functions is in the regulation of breathing, where TAS2 senses pH changes that are correlated to changes in the concentration of carbon dioxide in the blood, to determine the firing rate of retrotrapezoid nucleus neurons that instruct respiration muscles, and determine the rate of breathing. So this is another small membrane protein. This is about 60 kilodaltons of ordered membrane protein. This is a 2-pore domain family ion channel in which 2 of these canonical potassium channel pore domains are concatenated into a single chain. And so we were interested in understanding how protons regulate the activity of this channel and set out to determine its structure. This is work of another fantastic postdoc in my lab, Balvin Lee, who's currently on the job market, in collaboration with a graduate student, Robert Riedmeyer, in my group. And so Balvin was able to purify TAS2, reconstitute it into lipid nanodiscs, and determine its structure at 3.5 angstrom resolution using data collected again with Dan and John here at UC Berkeley on our Arctica microscope. And so this structure and another structure at a lower pH allowed us to understand new aspects of how this channel is regulated by protons. And so Balbin and Robert found that task 2 exhibits 2 new kinds of channel gate from these 2 structures, both at around 3.5 angstrom resolution. One of these gates is near the extracellular aspect of the channel and involves conformational changes to the selectivity filter, which forms potassium coordination sites that determine the selectivity of this channel for potassium. The second gate is at the intracellular aspect of the channel and involves a rearrangement that pinches this region of the channel shut. So similarly, we were interested in whether we could understand more about how this channel operates from higher resolution structures. And so Balbin, in an analogous way, took the grids out of our Arctica that we didn't collect on, shipped them to the Netherlands, and Abhay was able to collect a data set for us. And these data led to a reconstruction that was again improved substantially from our initial reconstructions, this one to 2.5 angstrom resolution. And I'm showing the final map here on the right of this slide. And I just wanted to point out 2 aspects that we were able to model that we were not able to model in the lower resolution reconstruction. The first is, again, portions of these membrane scaffold proteins that form the belt around the lipid nanodisk. And in this case, similar to the case of 3A, make some specific interactions with residues and transmembrane helices on the channel. And the second aspect are a number of ordered lipid molecules, several of which I've pointed out here. And I'd like to draw your attention to one of these lipid molecules that we can see in the higher resolution map and that I've colored in red on this slide here. And I'm going to show you a view from the cytoplasmic aspect of the channel up towards this lipid to illustrate why we find this particular lipid very interesting functionally. And so this lipid, as it turns out, is bound inside the channel cavity through that kind of lateral membrane opening. And in this position, it sits directly underneath of the potassium selectivity filter. And in this position, it would appear to block conduction. And we find this really quite fascinating because there is evidence from our work on related channels that this may be a conserved means of regulating channel activity in this family by having lipid block of ion conduction in this conformation in which lateral membrane openings are exposed. And so what we can see in this slide here is that from the lower resolution reconstructions, we were able to understand how protons are sensed on the extracellular and intracellular sides of the channel to gate the channel closed with the selectivity filter and inner gate, respectively. And we're now very interested in understanding how this potential lipid block of ion conduction works in these channels and how it interacts with these other proton gates on either side of the membrane. And so with that, I would like to acknowledge my lab and again, just point out that the microscopy that I talked about was done by David Kern with help from Chris Hole and Balbin Lee. And I would like to acknowledge my funding sources and collaborators, especially Abhay Kotecha at Thermo Fisher, Dan Tasso and John Remus here at our Cal Cryo-EM facility, Diana Bautista and her graduate students, Mali, and Hillel Adesnik and his postdoc, Savitha Sridharan, who are collaborators on the 3A project for functional work that I didn't talk about. So thank you very much.
Thank you, Steve, for this wonderful talk. It's really amazing to see because just a few years ago, talking about such sub-100 kilodalton proteins and cryo-EM was very difficult to imagine. And now you showed that we can not only work on such proteins, these proteins were very flexible and also very heterogeneous. and yet you can get such high resolution. This just shows that how quickly in how such a short time technology has come together, which allows you to answer very difficult biological questions. So what I'm going to do now is spend a few minutes on cryos first, and then go on to talk about Glacios, Selectris, and Falcon 4 as well. So first, as Mark Strom said earlier, the power of 4, that's a Cryos G4, the EC-FEG, Selectris, and Falcon 4. Here, what we did was, around the end of last year, in December, we did this very nice project. This collaboration was initiated by Tomohiro and Masahiro at University of Tokyo. They were working there on streptavidin. Streptavidin is a very small 52 kilodalton protein. It's very flexible and very heterogeneous. In 2019, Hongwei Wang showed for the first time that they can obtain a high-resolution structure going to 3.2 angstroms using cryo-EM. They use faceplate at the time to do this. About 6 months later, another group from Princeton University, Ning Yang's group, pushed the resolution further to 2.6 angstroms without using the faceplates. End of last year, Tomohiro and Masahiro started testing this streptavidin on their new Cryos G4. And this is how it looked like. So this is the grid on which they collected the data. These were the areas, grid squares, where data was collected using G4 at Tokyo with bioquantum K3. And from this, they obtained this beautiful structure of the streptavidin going to 1.9 angstrom. This is already beautiful because this is the first structure of a sub-100 kilodalton protein going to sub-2 angstrom resolution. What we did next was even more exciting. They took this grid out from their autoloader in their lab in Tokyo, and they shipped the same grid to Netherlands, to me. And what we did is, using my microscope, the Cryos G4 with the CFEG, Selectris, and Falcon 4, I collected the data in the similar grid squares near where they had collected the previous data set. We tried and made sure that we keep as many parameters as possible constant. So in terms of pixel sizes were similar, in terms of dose was kept similar, the defocus range was kept similar. It was a different microscope and has a Selectris and Falcon 4 detector on it. And then this is how the data looks like. This is one of the image of streptavidin. It's low pass filtered to about 20 angstroms, so we can clearly see all the particles here. And Masahiro went ahead and processed this data. And with similar amount of particles, he produced this beautiful 1.7 angstrom structure from the same grid. Now this is even more exciting, that we can further even after the grid was sitting in the microscope for some time and was transported published a paper with LMB Cambridge on atomic resolution structure of apoferritin, as well as 1.7 angstrom structure of a human GABA receptor. There, with the GABA receptor, we saw that at 1.7 angstrom, we can actually see hydrogen atoms. So then the question was, can we really see the hydrogens even here with 1.7 angstrom data? This was done in collaboration with people at LMB Cambridge, especially Kietaro, who works in Garib Mashudo's group. And indeed, we can actually see hydrogens in this difference density here. All these little sticks that we see, as well as this green blobs, these are the densities for hydrogen atoms. So not only we can push the resolution of such a small protein to 1.7 angstrom, but now hydrogen atoms are also visible. Again, showing how far this technology has come in a very short period of time. So this is how we have been working so far. These are all the structures that we have derived using the cryos with Selectris and Falcon 4. I already showed this 1.7 angstrom streptavidin. Last year, I also showed you hemoglobin structure going to 2 angstroms. Steve gave a wonderful talk and showed that even with membrane proteins, challenging membrane proteins, because these were very flexible and very heterogeneous samples, we can push the resolution to 2.1 angstrom in case of this 3A ion channel, as well as 2.5 angstrom for the human potassium channel. Again, showcasing what we can do nowadays, even with such small proteins and cryo-electron microscopy. Moving further, we have this wonderful collaboration with a group of Radu Arasescu at MRC LMB as well in Cambridge, where they are interested in looking at structure-based drug design. Radu is working on a human GABA receptor. GABA receptor is a 200 kilodalton human membrane protein found in our brains. It's acting as a neurotransmitter, and it's responsible for our moods, depression. It's also controlling anxiety, epilepsy, insomnia, and it has been a target for all these diseases. In addition, it's also the drug target for things like sleeping pills, as well as anesthetics. So it's a very important receptor. It's an asymmetric protein, forms a chloride ion channel, and there are lots and lots of drugs against this channel. One of the problems is this channel could not be crystallized at all after decades and decades of work. Only way to get structures is by cryo-electron microscopy. So we looked at this sample, and again, we can see these wonderful results on a bottom-mounted Falcon 3 or Falcon 4. For these 3 different drugs here, we can obtain 2.6 angstrom structures, where you can clearly see how this drug is bound. And in case of 2 of these drugs, we managed to put them onto Selectris Falcon 4 system. In case of this RO154513, which is a drug against alcohol intoxication, we used exactly same grid, which went onto the bottom-mounted Falcon 4 first, and then went onto the Selectris Falcon 4, and the resolution was improved to 2.2 angstroms. And in case of zopiclone, resolution was improved even further to 1.9 angstroms. We also looked at more drugs. Here I'm showing you 2 more examples. Alprazolam, which is a drug against anxiety as well as panic disorder, going to also 2.2 angstroms, and diazepam, the drug against seizure, going to 2.5 angstroms. So now we have like this range of structures with all these different drugs bound within this GABA receptor, and Radu's team is working on trying to understand the function of this and use this structure to do some rational design to make the drugs better, to reduce the side effect, to just improve its capabilities. So this is what we can do with cryo-EM, with cryos and Selectris and Falcon 4. And now what I would like to show you is spend a few minutes trying to show what we have been doing with the Selectris and Falcon 4 also on a 200 keV Glacios system. On the Glacios, so here we looked at proteasome T20S. This is a collaboration with a group of Jürgen Plitzko at the Max Planck Institute. On unfiltered images with Falcon 4, so like kind of simulating a bottom-mounted system, we can see particles when we go further away from focus. So here about 0.8 micron defocus, we can start to see that yes, particles are present. But when we are closer to focus at 0.4 micron, particles are hardly visible. As soon as we do the filtering with the 10 EV slit, in this case, the contrast improves miraculously. As you can see, like very close to focus as well, all of these particles are visible. And it's not just the contrast, it's also the signal to noise that's improving in these images. And since you have better signal to noise, particles are aligning better, and this leads to higher resolution. And this is exactly what we observed here. With unfiltered particles, we can get about 2.34 angstrom structure. When we use a 20 eV slit, resolution is further improved by about 0.08 angstroms, going to 2.26. And when we use even narrow slits, so now we are removing all the noise from this plasmon peak, the resolution is further improved to 2.14 angstrom. That's a massive difference, going from 2.3 to 2.14. It also shows that the 20 eV slit, which is generally used standardly in the field, it's good, but with 10 eV, you can get even better resolution. Now, it's about 0.12 angstrom difference between 20 and 10 eV, and about 0.08 angstrom difference between unfiltered versus filtered imaging. Is this significant? If we look at the B factors, then we can see that yes, there is definitely a big change going on from unfiltered versus filtered. Now, all this data was collected on exactly same grid, keeping as many parameters same as we possibly can. And it was also collected consecutively within 1.5 days. Each of this data set took about 12 hours. It was running at about 250 movies per hour using ARFIS. So we try to keep as many things same as possible and still can obtain such high resolution structures. We can also look at this by plotting these B factors. So here I'm plotting the B factors in an exponential form. And what we can see is this green line belongs to the 10 eV slit, 20 eV is the yellow line, and the unfiltered is the red. And there is already a difference between these data sets. If we were to just take a point on this plot, let's say 2 angstroms, how many particles we would need to reach 2 angstroms? Then it becomes clear that with 10 eV slit, we will need about 370,000 particles of this type to reach 2 angstroms if we use 10 eV. If we were to use a 20 eV slit, then we will need 1 million particles to reach the same resolution. And if we don't use the slit at all, if we use bottom-mounted Falcon 4 without any filtering, then it would almost become impossible for this data to reach 2.1 angstrom or 2 angstroms in this case. So indeed, selectress or energy filtering is helping you tremendously, even at 200 kV as well. Now, this was all done with proteasome 20S, which is a 700 kilodalton protein and has a D7 symmetry. Next, we wanted to see what happens if we put something more challenging and something smaller. So we tried this on a GABA receptor again. This is a homopentameric form of GABA. It has a 5-fold symmetry. It's about 200 kilodalton protein. And there we can see the difference is even bigger. With unfiltered, we can get 2.8 angstrom resolution. With 10 eV, the resolution is improved to 2.4 angstroms. It's like a difference of 0.4 angstroms, and also with less number of particles. And again, everything was done using the same grid and same parameters between the two sets. So once again, it shows that filtering is really helping push your resolution for single particle analysis. And at this resolution, you can already see your bound drug molecules. You can see all the ordered water molecules, and can be used for structure-based drug discovery. Now, this has C5 symmetry. We wanted to see what happens if we put even more challenging sample which doesn't have any symmetry. For that, we tried to look at a GPCR sample. This is a collaboration with the group of Patrick Saxton at Monash University, as well as Radostin Danau at Tokyo University. Here we looked at GPCR sample GLP1R. Another thing we did was we wanted to like push even further and see what happens if we try and look at sub 100 kilodalton proteins. For this, we have another collaboration, and this collaboration is with Basil Graeber at the Institute of Cancer Research in London, where he's working on this human CDK activating kinases, the CDK cyclins. He sent us this sample. Now, this sample was very challenging because not only it's very flexible, it has very high heterogeneity, but also a significant portion also has preferential orientation. However, with Selectors and Falcon 4 on the glaciers, we managed to find a population where there were all the orientations present, and the structure was pushed to 2.5 angstroms. Again, showing that what we can do even with a sub 100 kilodalton. This protein is about 85 kilodalton in the aligned region. There is also some flexible bits, about 15 kilodalton, which is not visible in cryo-EM map because it's highly flexible. So also with the glaciers, we have seen a range of different structures, starting from apoferritin going to 1.7 angstrom, the T20S to 2.1 angstrom, this 85 kilodalton CDK activating kinase to 2.5 angstroms. We also looked at the hemoglobin, which is a 64 kilodalton protein, which went to about 3.4-ish angstrom resolution. And in case of membrane proteins, we looked at the GABA going to 2.4 and this GPCR sample going to 2.6 angstroms. With this, I would like to thank all our collaborators for this wonderful support throughout this work and also contributing to this really nice samples and all their expertise. And I would also like to thank you everyone for listening to this webinar. Thank you.
Well, let me start with the first question. So I think I'll ask this one of Abhay. What resolution do you need for meaningful interpretation of for drug discovery?
Sorry, Leah, there was a lot of echo in the beginning. Could you repeat the question? I just heard my name.
No problem. What resolution do you need for meaningful interpretation for drug discovery?
Okay. All right. So for it actually depends on what question you are asking. If you just want to see whether a ligand or your drug molecule is present in the binding pocket or not, then you don't need very high resolutions. One could get this information already at around 3.5 angstroms, in some cases even around 4 angstroms. If you want to start identifying how this ligand is interacting with the surrounding environment, then you need higher resolution. Once you are at 2.5 angstroms, you start to see how this binding is happening, what all the hydrogen atoms are there. You can model the ligand properly as well as all the side chains in your binding pocket. And as soon as you start going higher and higher resolution, like going from 2.5 to about 2.22 angstroms, you start to see all the water molecules. And water molecules play a very important role, especially when you want to do like a structure-based drug discovery. So I would say like anywhere between 2 and 2.5 angstroms, you start getting more accurate details. From 2.5 to 3 angstroms, you can still see that how your ligand is bound and how it's interacting with little less accuracy. And from 3 angstroms to 4 angstrom, you can do some predictions there.
Great.
Would you agree, Steve, with this? Steve, can you hear me?
Yeah, sorry, just didn't catch that. Yeah, I think that's exactly what I would say as well. More information, you can learn more about the details of these interactions, but yeah, that makes sense.
Okay, thanks. So in terms of maybe resolution, so how much do you think the higher magnification on the cryos had on the final resolution of these maps? Abhay or Mark, would you like to answer?
Yeah, I can at least say in our case, we don't know. We haven't done a comparison with the exact same pixel size on different microscopes.
Okay. And I think it's maybe a specific question to your Task 2 project for you, Steve. Have you tried modulating different lipids for the nanodisk, such as lipids unique to the brain or other organs?
Yeah, that's a great question. We have not yet. We'd be very interested in doing that, especially with the higher resolution structures now, where we can really make out what some of these interactions look like.
Okay, thank you. So then I think question for Abhay, was the streptavidin dataset also collected with the Colfeg, or only Falcon 4 and Selectris?
The streptavidin data was collected with the Colfeg, Selectris, and Falcon 4.
Okay. And given the same conditions, sample grid detector, what would be a typical difference in the overall resolution range expected observed in the data set collected on a cryos and glacios?
That depends. We have done some experiments with T20S and GABA sample. I showed you some T20S data. With Selectris and Falcon 4, we can achieve this 2.1 angstroms on the glaciers. Same sample, it was not exactly same grid, it was a different grid, but same sample, same kind of ice thickness. When collected on the cryos, we can also, cryos with a bottom-mounted camera first, we can also achieve 2.1 angstrom, however, with less number of particles. I think it was almost half the number of particles there. In case of GABA receptor now, where there is a lower symmetry, some more flexibility, some more challenges, there the difference starts to become slightly bigger. On the glaciers with Selectris, we get 2.4 angstrom. On cryos with a bottom-mounted Falcon 4, we get to 2 angstroms. And cryos with Selectris and Falcon 4, we push it to 1.7 angstroms in case of a symmetric form of GABA.
Okay, thanks. Maybe just a quick follow-up question related to a few questions about Selectris. So have you tested the benefit of the Selectris on samples with thicker ice or tilted samples when the resolution is usually limited?
Not really like a direct side-by-side comparison where I look at the same sample on a thick and thin ice. But in principle, when you have a thicker sample, you have more inelastic scattering. Filtering should help even more. But it is a very good question, and we should do some experiments on this. In theory, yes, it should help, and this is the whole basis of tomography. That thicker sample filtering is going to help you much more.
Okay. So we also have a few questions about the nanodiscs. So with the nanodisk results without the proteins, were they present in solution? Steve, that's for you.
Oh, when we purify samples in nanodiscs, typically there's some portion of the sample that are empty nanodiscs that are going to be frozen along with the target in nanodiscs, and we just need to remove those computationally as we process data.
Okay, and maybe same question. The concentrations of the nanodiscs, were they too high, or?
I think they worked quite well. So that's actually how they do look high. We like to have the particles tiled quite closely on the grids. We find that it gives us thinner ice and makes processing easier, actually. So that's about where we like them to be. In those two cases, they're between 1 and 2 mg per mil for the 3A project and the test 2 project. But that's quite sample dependent, what concentration we need to go to have distribution that looks similar to what you saw on the slides today.
Okay, great, thanks. So based on your experience, what are the suitable detergents for membrane protein cryo-EM grids? And how did you overcome the difficulties caused by the detergents? Maybe Steve or Abhay, I don't know if you want to answer that first.
Sure. I tend to prefer to be in nanodiscs, and so that's how we avoid the problem. But Abhay probably has more experience with other detergents.
Indeed, I mean, this is why we want to move everything towards nanodiscs. It was the same with GABA as well. All the GABA work that I showed, the sample was within the nanodiscs. When Radu's group tried in early days with detergents, they saw lots of differences in how this channel was behaving, because the channel is very dynamic. In most cases, the channel was found to be collapsed. So yeah, generally, if you can, you should move away from detergent, try to go towards nanodiscs with lipids from similar environments if possible. But if there is no other option, you can always try with DDM, for example. That's the first detergent that I have been trying. Now there are even better detergents. LMNG, for example, has been shown to work very well with membrane proteins. And there are also several detergent screens already available, which you could try. But again, I would say the same thing, that if you can use nanodiscs, I would go with nanodiscs instead of going through the detergent route.
Okay. All right, thanks. Maybe along those same lines, question was asked, like, for the resolution improvement of these cases, how much can you attribute of hardware, and how much is attributed to protein preparation specific conditions?
The sample prep is always the main thing. In terms of hardware, with using standard samples such as apoferritin, for example, we can test our hardware very well. And last year, we showed that using the same system, this G4 with C-X selectors and Falcon 4, when we use apoferritin sample, we could really get to the atomic resolution, and we could get 1.2 angstrom with the best B factors ever. So we know that, yes, hardware is good. Now we have to work towards optimizing the samples, and it happens in two things. Like, you need to have optimized grids first, and there are lots and lots of improvements happening on the grid side. More recently, we saw this wonderful grids from Chris Russo, where they use hexagonal forms. Now also, Chris and Lori a few years ago showed that the dual grids are much better, and we also see the same thing. The dual grids do behave better in terms of this motion compared to the carbon grids. So that's one thing. Second thing is your ice quality. Of course, thinner ice the better, but at some stage you start having problems with air-water interface if you try and do much thinner ice, or even ice starts melting in some cases. And then also sample. Sample has to be taken. If we put very flexible, very heterogeneous sample, then we are not going to get very high resolutions. So again, some biochemistry helps there.
Okay, thanks. So you started talking about the hardware. I'll switch there for this question. Will the ECFEG also be available for retrofit possibilities on existing Cryos microscopes? Mark?
Yes, Leah, that is indeed the case. So if you have Cryos of the generation G3 or G3i, and you upgrade the instrument to Windows 10, then you can indeed have the CollFEG retrofitted onto those instruments.
Okay.
And of course, in addition to that, the Cryos G4s, of course, can always be equipped also as a retrofit later on in the field if you decide to add on a CollFEG later on.
Okay. You also talked about the flashing interval that is fully integrated into EPU and Tomo software. But when is such a flashing procedure triggered, and what is the interval between the flashing generally? Mark, I think that was
so typically, actually, when the beam current drops 10%, then you get actually a trigger of a flashing procedure. In the Nature paper of Nakano et al., we have actually reported that such interval is roughly 8 hours between the flashes, but we have also seen on our internal instruments that we can easily reach intervals which are more than 10 hours, so really actually a long time.
Okay, thanks. So I think this will be our last question. This one will be, I think, for Steve. Was there a difference in the data processing of the small proteins with the Cryos data? Was it difficult to process the data for such small proteins?
Yeah, it's a good question. They were difficult data sets to process. That's for sure. I think that, you know, it is a testament to how talented David and Balvin in my lab who process this data and Chris Hull are, that they got these things to work. We did not, though, see, you know, if anything, it was maybe a little bit easier to process data collected with the Selectris on Abhay's microscope than it was to process data from the Arctica. But both use similar pipelines, and you can kind of check out the details in the papers and the preprint, and those use a kind of approach that we found to be pretty robust for a few projects of similar size targets.
Okay, great. So I think with that, I want to thank you again, Steve, Mark, and Abhay, for your time today and for answering questions, as well as sharing your important research. Before we go, I'd like to thank the audience for joining us today and for your interesting questions. Questions, unfortunately, that we didn't have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. If you are a current customer and would like more information, such as a data sheet, please contact your account manager. If you don't know who that is, please contact us in the forms and the links provided in the resource window in the LabRoots module. This webcast can be viewed on demand. LabRoots will alert you with an email when it's available for replay. We encourage you to share that email with your colleagues that may have missed today's live event. Please join us again on May 19th for our next webinar on our next Thermo Fisher Electron Microscopy monthly series on LabRoots. You'll be hearing about our new capabilities for correlative microscopy and our integrated light fluorescence microscope that is part of our electron cryotomography workflow. The registration is open. So again, thanks again to our speakers. Thanks for you all for joining us. Until next time. Goodbye.